In the second part, we are going to talk about the scientific method. Before we go into that, we have to look at some of the assumptions that are necessary in order to use the scientific method. The first of these is the fact that the Scripture describes the origin of all things, but also furnishes us with a framework which we can interpret and view life in the world. This is probably one of the most important things that I will say in these videos for this lesson. God has created the world with consistent laws which are always true. What I mean by that is, if you look at the Greek god system, they were essentially superhumans with human emotions and moods, and so they would affect creation and nature in a very inconsistent way. But our God has created the world in such a way that we can depend on it to be the same every time. If you throw a rock out of the window, it's going to fall to the ground. There are rare exceptions to these rules. Most of these would be called miracles of nature or in nature, but for the vast majority of things, the consistent laws are always true. That said, there is a cause or reasonable explanation for every effect that occurs in the universe, and it reliably happens time after time after time. Lastly, the Scriptures are such that it says that there's a reality of the world that can be discovered and understood. So, this leads to a scientific method for solving problems or scientific problems. The scientific method is a systematic process for scientific investigation, and this is a form of inductive reasoning. As you know, there are two forms of reasoning: inductive reasoning and deductive reasoning. Deductive reasoning goes from a general principle and you deduce specific events or things that happen. Inductive reasoning, you look at a series of specific events and you hypothesize or talk about a general cause for those events. The scientific method is a way of systematically invoking inductive reasoning. Now, in the midst of performing these sciences, you have to realize there is a rule of conduct which applies to the scientists that do this. And this rule of conduct is called scientific ethics. For example, in order to perform an experiment on an individual, it's required that you gain their permission. In order to perform experiments on animals, there's almost always a ruling body at the university or scientific place where the scientific experimentation is being performed that you have to pass through in order to make sure that you're not unnecessarily cruel to the animals involved. So what are the steps in the scientific method? The first step is to state the problem that you want to answer clearly. So you state the question very clearly. Secondly, you develop a list in your mind, or on paper if you will, of the possible solutions that can occur, and then you pick one. And that single solution is called the hypothesis, or the educated guess that solves the problem that you've clearly stated. Then you have to test this hypothesis or these hypotheses that you've generated, and you do that performing what's known as a controlled experiment. A controlled experiment is a process where everything is kept constant in the experiment except for the factor that you're interested in. The factor that you're interested in that's manipulated is called the independent variable. The factor that you think is going to change because of the manipulation that you've done is called the dependent variable. So you observe the dependent variable for changes in the independent variable. The group in which you exercise the change of the independent variable is called the experimental group. So that's a group of individuals, a group of animals, a group of plants, a group of people, in which you're going to make a single change, keeping everything else the same, and you're going to see if how the dependent variable changes. And many times you have what's known as a control group, and this is a group where you don't change anything, and you compare the dependent variable outcome in the experimental group with the dependent variable outcome in the control group. And then generally, at the end of your experiment, when you decide you want to move forward, you frequently do several trials of changes of the independent variable, looking at changes in the dependent variable. And this is called replication. You can also perform the entire experiment over again, and that's also called replication, or looking for the same results. After you look at the changes in the dependent variable, when you change the independent variable, you reach a conclusion. And the conclusion is you either accept, the conclusion that the way that scientific method is set up, you want to reject what's called the null hypothesis, which is you take your hypothesis and you state it in a negative manner, such that you say, this is not the reason for this change. And then you develop enough evidence that you can reject that null hypothesis. Once a series of experiments has been done, a hypothesis can progress to something known as a theory. A theory is a hypothesis that has a reasonable amount of support for its conclusion. And a theory can then become a law. A law is a theory that has been out there for a period of time and still looks like it's true. So, for example, you can start out with the theory of gravity, or you can start out with the hypothesis of gravity, and then after a series of experiments, you say, well, it looks like gravity is real, so it becomes the theory of gravity, and then after another period of time, it still seems to be true and real, and it becomes the law of gravity. Now, let's look specifically at how the idea of spontaneous generation was reputed using the scientific method. And there were two individuals that were much involved in this. One was Francisco Redi, who did a series of scientific method related experiments to disprove spontaneous generation, as did Louis Pasteur, who also proved that living organisms could not come from non-living. And they developed the law between them. They developed enough evidence that it's now called the law of biogenesis, which means that only living things can come from other living things. That things that are not living, matter that has no essence of life, is not able to produce life. So this is a picture of Redi's experiment. What Redi did was he took a piece of meat and put it in a jar, and the first piece of meat, he left the jar open, and as you can see, the flies flew in and out, and what happened after a period of time was that there were worms all over the jar. Then he put a strong material over the top of the jar and shut it tight, and he showed that the flies could not get in, and there was no worms or maggots on the meat. Now, in order to show that it wasn't because you were excluding air from the jar by the tight seal, he covered the jar with gauzy material. And interestingly, when he did that, the flies came and landed on the gauze, but they couldn't get to the meat. And the maggots then formed on the gauze above the meat, but not on the meat itself. So he proved, by doing this, he proved that the meat was not the source of the maggots, but the flies were. And he also proved that even if the air and all the other materials that necessary to support life were available, that even with all that, the meat could not produce life. Now, what Louis Pasteur did is he had a series of flasks. And these series of flasks, the first he heated, and then he let the flask sit, and no bacteria developed in the flask. And he put this very long curved tube on it so that he could say that air was able to get in and out of the flask. Okay, but he made it long and curved so that if there were any particles or anything in the air, that they would be trapped in the tube and only real air could get in and out. No bacteria is present. So then the people complaining about this, they said, well, when you heated it up, you killed the life essence in the material, so it wouldn't support life. So then he after a period of time, and he broke the neck of the flask, exposed it to the air this way, and bacteria formed. And then, in order to show that it wasn't specifically because of how he broke the neck of this neck, he took the same and tilted it in such a way so that the material could go up into this S-shaped area, and then he tilted it back, and he showed that whatever was causing the life was actually trapped in the neck. And so these experiments together showed that you couldn't produce life from non-living things. You had to expose the various situations to living things in order to... Now, the limitations of science. Scientism is the belief that science can answer all of life's problems, and that's actually not true. There's the problem of sin, which science cannot particularly address, sin and selfishness, etc. Biology is essentially limited to being able to investigate and find God's design in the physical universe and apply that knowledge to help man dominate the earth. A personal example, we were doing a seminar, I was doing a seminar in a scientific situation, and basically we were talking about something known as the CDA cycle, or the cyclin D2 cycle. And this individual, and we were having a conversation, this individual was described as the guy who created the cyclin D2 cycle. And I said in that conversation, I said, he didn't create it. He just found it and described it. And that's what science does. It finds what's already there and describes it. Just because a person can describe how a rainbow happens doesn't mean that it wasn't created the way it is. So let's look at a couple of examples of scientific method. First of all, does the number of coils of wire in a magnet affect its magnetic strength? You can make an electromagnet by wrapping wire around a piece of metal, like this wire around this nail. The question here is the number of loops, does that impact the strength of the magnet? And how would you test that? Well, first of all, the question is, does the number of coils in the wire affect an electromagnetic strength? The hypothesis is yes, the number of coils does impact the magnetic strength, or the null hypothesis is the number of coils does not impact the magnetic strength. You would then create a series of several magnets, and the only change between each electromagnet is the number of coils. The independent variable, the variable you change, is the number of coils. The dependent variable is the magnetic field strength. And as it turns out, you don't need, in this case, you don't need a control group that these act as, the original magnet can act as the control group for the series beyond it. The conclusion, if you do this experiment, is indeed the number of coils does impact a magnetic field strength, or you can reject the null hypothesis that it does not. And this formula shows Ni, this is the magnetic field strength, this B. Ni is the number of coils. And so the fact that you're multiplying these factors, the larger N is, the greater the field strength. So clearly the number of coils does impact the field strength. A second example is, does the weight of a pendulum, or mass of the pendulum, mass of this swinging object known as the bob, does that mass impact its period? The period is the time it takes the pendulum to swing through one complete swing. So from maxima here through to maxima here and then back to here. That time is called the period. So does the mass of this ball have any impact on how long it takes this ball to get one full swing? Again, the question is, does the weight of the bob or the ball affect the period or the time it takes to make a full swing? The hypothesis would be the weight of the bob on the pendulum does impact the period, or the null hypothesis is the weight of the bob does not, on the pendulum, does not impact the period. A control experiment would be similarly to make a series of pendula whose only difference is the weight of the bob. So everything else, the length of the swing, etc., would be kept the same. The independent variable is the weight of the bob. The dependent variable is the period, or the time to swing through a complete swing. Again, in this setting, the original pendulum can be your control, and all the new pendula that you make are the experimental groups. And in this case, you actually accept the null hypothesis, because what you see as you do the experiment is that no matter what the mass of the bob is, the period remains the same. And here's the formula for pendulums, where T is the period, L is the length of the pendulum, and G is gravity. And what you notice that's not in here is the mass. And so the mass has no impact on the period of the pendulum. And if you remember, if you have a grandfather clock, that in order to change the period swing, you don't change the weight. The weight remains the same, but you slide it up and down on the swing, either lengthening or shortening the swing.